Assalamu alaikum. Assalamu alaikum. Today we are going to discuss the continuation of the previous lecture, that is transport across the cell membrane. Now we are going to discuss osmosis. What is osmosis and how we can understand the concept of osmosis. Actually, osmosis is just a movement of water molecules through a selectively permeable or semi-permeable membrane like cell membrane. Water diffuses across a cell membrane from an area of higher concentration to an area of lower concentration. So it is a movement of water through semi-permeable or selectively permeable. Selectively or semi-permeable. I should do this to this. Selectively permeable. Now, what about the semi-permeable membrane? Semi-permeable, I would write in a short. Semi-permeable means that the membrane that is permeable to water but not to solutes, that is sugar, only water, not solutes. For example, sugar. So I took the advantages of to edit to pause the video. I have already drawn some diagrams to understand you when there is a water that is generated by blue and the sugar that is generated by this color, and this is selectively permeable membrane that is um, only allow the water to pass. So this is the higher concentration, this is the lower concentration. So the water move from this side to this side. So the water will move from higher concentration towards the lower concentration, but the solutes remain the same. If we look into another diagram, which I've already drawn by pausing the video. So in the first diagram, there would be a semi-permeable membrane. I will denote it with SPM, and these are the solutes. And the water will move from this higher concentration towards the lower concentration. In that area, there is less concentration of water, but sorry, less concentration of solutes in that area and higher concentration of water in this area, higher concentration solutes and less concentration water. So eventually, if you go to the other part, the water has been moved from this to this, and there will be a higher concentration of water, and this movement will still going on when there is an equilibrium achieved when the both sides, side A and side B, having the same amount of water. But the selectively permeable membrane, that one is doesn't allow any sort of movement of solutes. Now we should move towards the tonicity. So what is the tonicity? Tonicity means that the solution is hypertonic, isotonic, or hypertonic. That is mainly by the solutes or normal saline or sodium ions. If the movement of solutes is less, then there is an hypotonic. If equal, that is isotonic, and if it is more, then it is hypertonic. Or we can understand in a simple way, we have cell. This is extracellular fluid compartment, this is intracellular fluid compartment. If the amount of sodium ions or solute is less than inside, I should change the color, 3, then it is hypotonic. If the amount of sodium ions is equal to the inside of the cell, that is isotonic, the same composition of the fluid that is having sodium ions equally distributed in extracellular fluid and the intracellular fluid. But if the amount of sodium ions greater than the amount of sodium ions inside the cell than outside the cell, then it has become hypertonic. So in daily our medical practices, we need to know very much about the fluid concentration and their tonicity, because some patients needs hypotonic solutions, some needs isotonic solutions, and some needs hypertonic solutions depending on their medical diseases. Now we'll discuss separately about this tonicity of the fluid. We first discuss hypertonic. If we made a cell like this is intracellular fluid, and if we put in a medium containing solution, that is hypertonic, it means that having solutes more than the intracellular fluid or sodium ions. Actually, hypertonic solutions contain a high concentration of solute relative to the another solution. So this fluid, this one, is having high concentration of sodium ions or solutes. When the cell is placed in such hypertonic solution, the water diffuses out of the cell and causing the cell to shrink. So this cell membrane of the cell, this cell membrane of the cell work as semi-permeable membrane. It diffuses, or it allows the water to move outside the cell, but not the solutes. We already know and equally know that there is a solute concentration higher in outside the cell than inside the cell, but this selectively permeable membrane does not allow the movement of solutes or sodium ions. So the water diffuses out from the membrane and the concentration of the solutes remains same as previously. So this will causes the cell to shrink down. This will causes the cell to shrink down. It will squeeze the cell because it has been drained all the water from the cell. This is an typical example of hypertonic. Now we would go for an hypotonic. The same is the case if we put a cell in a solution, cell membrane, okay, we donate like this. This is the cell having in a solution of containing sodium ions less than the inside cell. This is the cell having sodium ions more as compared to the extracellular fluid. Hypotonic solutions contain a lower concentration of solutes rather relative to the other other fluids. So this is the selectively permeable membrane. So eventually, when the cell is placed in such an hypotonic solution, the water from the outside will move inward and making the cell to swell and nearly to explode. So if we give a wrong fluid, a wrong solution, a wrong drip to a patient or IV solution to the patient, opposite to its medical treatment or medical condition, it may worsen the condition. If the patient having solutes inside the cells more than the solutes outside of the cells, the water will move inside the cell, make the cell swell, and it will burst out. If patient is already dehydrated and we are giving him or her a hypotonic solution, then it will eventually adverse the condition and make the cell to burst out or swell out. So it eventually not good for the patient. So in hypotonic solution, they contain lesser or low concentration of solutes rather than the inside the cell. When the cell is placed such an hypotonic solution, the water diffuses from outside the cell from extracellular fluid to intracellular fluid and making the cell to swell up and explode. Now what about the isotonic? Isotonic means same. If we take another beaker, we put a cell inside having the same number of solutes. I would put this 6 ions for example and 3 here, and the cell is placed here. So there will be no net movement of the water from inside or outside. There will be a already maintained equilibrium between the extracellular fluid and intracellular fluid. Because the isotonic solution contain the same concentration of solutes as another solutions or as inside the cell. When the cell is placed to such an isotonic solution, the water diffuses into and out of the cell at the same rate, as I denoted by this. The fluid that surrounds the body of cells is isotonic, and there will be no shrinkage of the cell and will be no busting of the cell or swelling of the cell. So this is an example of isotonic fluids or isotonic solutions. Now I would give you some examples how these fluids are clinically used. Number one is isotonic, the same solution, the same solution with the composition from inside the cell as compared to outside the cell, that is 0.9% sodium chloride, dextrose in water. Number two, hypotonic. Hypotonic, it eventually or it is used for mainly to treat dehydration, Dehydration may be due to gastroenteritis, vomiting, and diarrhea, or may be due to blood loss. And hypertonic solution. Hypertonic solution is mainly used to treat the cerebral edema. For example, if the normal size of the brain cell is this, and it will become like this. So by giving the hypertonic solution, it will squeeze out the extra water from the brain cells and eventually make it to this size, normal size. Due to any medical condition, these medical conditions will be taught you by in your clinical scenarios or in your clinical classes. That would be eventually help to understand the basic concept of this tonicity of the fluid. But this is very much important, as this is the basic uh, this is the basics of your medical sciences, and you will have to deal with this throughout your life. You have to treat patients, you have to know about the hypertonic, isotonic, and hypotonic solutions to give in a certain specific indications. Not every person is candidate for hypertonic, not every person is candidate for isotonic, and not every person is candidate for hypotonic. Now we'll move towards the active transport. We already discussed or know from our background knowledge that active transport is is a transport that move against the concentration gradient, move against concentration gradient. That means that is moving from lower concentration towards the higher concentration. Number two, it required energy. Number three, and there is some mechanical or structural kinetics involved. Kinetics involved. For example, you have to push against the resistance. So it should there should be an energy needed, and the concentration gradient is already I had mentioned, and there is some kinetic energy or the mechanical energy that is used to push the specific type of molecules or solutes against the concentration gradient. Active transport is divided into two types according to the source of energy of the use. So, active transport is divided into two types. Number one, actually, it depends upon the source of energy to to cause the transport or to avail the transport. Number one is primary. Number two is secondary. First, we first we are gonna discuss the primary. So I change the color. Number one, MARY, primary transport. Primary transport mainly involve three active or mechanical pumps. Number one is sodium potassium pump, sodium potassium pump. Number two, calcium pump. And number three is the hydrogen potassium pump. These are the main pumps that is used in primary active transports. Now I'll try to draw the sodium potassium pump, and it's this is the cell membrane. This is outside the cell, extracellular fluid. This is the intracellular fluid. The energy basically used for this is in the form of an ATP adenosine triphosphate that is converted into ADP. So we draw this the same. We'll having these ions, any specific ions that having concentration more inside the cell as compared to outside the cells. These molecules tend to move outward due to concentration difference in normally. But if the concentration of move ions is more in extracellular fluid than intracellular fluid, then it is very much difficult for these ions to move these against the concentration gradient. So a pump is used by the requirement of energy called sodium potassium pump that will sorry that will move these ions against the concentration gradient against the concentration gradient opposite to the uh, against the concentration gradient with the expenditure of energy and forcing these low concentration molecules or ions into the higher concentration, and there will be an energy use in the form of an ATP that is converted into ADP, and the net movement is against the concentration gradient. This is a type of an sodium potassium pump of primary active transport. So what would be the functions of sodium potassium pump? Functions. I am discussing the main functions, not in detail. It actually maintains the sodium potassium concentration difference across the cell membrane. Concentration difference of sodium and potassium across the cell membrane. It maintains the volume of the cell. The cell is not going to shrink or not going to swell or explode. Volume of the cell, it should be maintained by this. Number 3, it actually causes an negative electrical charge inside the cell. Negative charge inside intracellular fluid. And number 4, it is very much important for use of oxygen in kidney cells or by the kidneys. So the second type is secondary active transport. Secondary active transport, it's you may call it is a partnership. Energy utilized in transport of one substance that would help the movement of other substance. That, or you can take a lift. If you are going somewhere and ask someone for a lift, the person is already going there, and it will also help on your side that you will also reach your destination by taking help of that person. So the energy is derived secondarily from the energy it has been stored in the form of ionic concentration difference of secondary molecule or ionic substances between the two sides of the cell membrane, actually created originally by the primary active transport. For example, if these molecules have to go there against the concentration gradient, this molecule also asks him, please carry with me. Actually, this one, the we should change the color. The yellow one will move by active transport, primary active transport. But this one, the other one, will use the energy of the first molecules and become secondary active transport. So the energy used by the first molecule is the primary, that is in the form of ATP, that is converted into ADP. But the second molecules acquire the energy of the first molecules, or taking lift from the first molecules, and become the part of, or become a type of active transport, that is secondary active transport. Now the third one is, so another form of transport is a co-transport. It's just like when you are taking lift from someone and going want to move your destination, the other person is already moving towards its destination, so you take a left from it and make it possible for you to reach your destination. For example, if we draw a cell membrane and having the carrier proteins that are mainly involved in this, so the there is an carrier protein and the sodium molecules, actually they using the energy and they want to go to inside the cell. This is extracellular fluid, this is intracellular fluid. But there is another molecule, that one is glucose, that is also want to go inside the cell, but it doesn't want to use any expenditure or energy or fare. Some of our friends do that because they don't want to use their resources. So this glucose will co-transport or attach with this sodium. These two attach, these move inside the cell along with the sodium, and glucose will move inside the cell using the energy of the sodium. That one is using already as energy of primary active transport, having ATP into ADP. So this is a co-transport or a lift transport that is done mainly by a carrier protein. The most common example is sodium glucose co-transport. Sodium glucose co-transport. Another example of transport is a counter transport. Counter transport means the transport of sodium, it via its concentration gradient, is coupled to transport other substances and outside the direction. The same example, this is cell membrane, this is extracellular fluid, this is intracellular fluid. If sodium wants to go inside the cell and some other thing or some other ions or substances wants to come outside the cell, so they use a counter transport channel. That other substances may be calcium, that is the most common example. So there is a protein here, sodium will eventually on attach to the this side, and calcium will eventually attach to the this side. So this molecule will twist like this. Sodium become inside and this calcium become outside against the concentration gradient. This is called encounter transport, or the movement of two ions through the cell membrane in an opposite direction. One is moving inside and another one is moving outside against the concentration gradient. Now we will discuss about its applied aspects. These transport or these counter transport are maybe used in cardiac muscles or cardiac glycosides, uh, or cardiac drugs that are called cardiac glycoside like digitalis, and that is used in management of heart failure, that inhibits the sodium potassium pump or the accumulation of sodium inside the cell, prevention of potassium influx, or intracellular accumulation of sodium, decreased sodium gradient from the outside to inside. That's all depend on their clinical aspects. Now we'll discuss about another transport that is called vascular vesicular transport. I should increase the size. It includes endocytosis, it includes exocytosis and transcytosis. Endo or exocytosis have already been discussed many times, or I try to draw the picture of that. My drawing is not so good, but I think you can understand about the concept. This is actually endocytosis in this way and exocytosis in this way. So, if molecules to be ingested, there is an invagination, and there would be twist, closing up the cavity of the membrane, and eventually the particles are engulfed. But if we want to exercise the substance, the particles or in a closed cavity, they should attach to the cell membrane, and then eventually it will be thrown out from the cell. This is very simple, and we have been studying in from um, from our childhood. In endocytosis, the material make contact with the cell membrane. The cell membrane then invaginates, and invagination is pinched off, leaving of the cell membrane in contact. Endocytosis has two types. Number one, receptor mediated, and number two is phagocytosis. Number three is pinocytosis. This is for solids, phagocytosis. This is for liquids. For example, white blood cells surround and engulf the bacteria, WBCs. These are done by the phagocytosis. Amino acids, and fatty acids. These are done by the pinocytosis and receptor mediated. nerve growth factors, vitamins, hormones, viruses, especially the HIV viruses that is engulfed by the receptor mediated phagocyt, uh, phagocytosis, receptor mediated uh, vesicular transport or uh, exocytosis or endocytosis. This endocytosis, basically these are the different types that has been criticized on the subtypes for the specific molecules and amount that that is being done in daily routine purpose in our body. Now about the exocytosis, I already mentioned you can this way. It is exocytosis. It is actually the process of release of macromolecules from the cells to the exterior. Vesicles containing materials may be exposed and bind to the cell membrane like this. An area of fusion breaks and leaving the cell contents outside the environment, and the reverse of it, it's pinocytosis or endocytosis. It actually requires an energy. um, it requires calcium for contraction and energy, and may require or may be needed some hormones depending on the body condition. For example, if we eat sugar, our blood glucose level will become high. It eventually lead insulin, that hormone that is produced in the beta cells of the pancreas, pancreas beta cells that are delivered into blood by the process of exocytosis, and they eventually the cells, and they will eventually decrease sugar, and this process goes depending upon your eating habits, your body metabolism, your stress level, your exercise level. Now, the other type of vesicular transport is transcytosis. It is somewhat like counter transport because vesicles endocytosed by the one side of the cell membrane, they will be exocytosed to the opposite side. Cavalian mediated rafts and Cavalli like mechanism. These are also called cytomapses, transport of substances across the cell membrane of the blood vessels of interstitial fluid. If having cell like this molecules has to be go inside and this molecules has to be go outside. So if this molecules is endocytosed and at the same time this molecules is exocytosed, this is transcytosis or transcytosis of the. Sorry for interruption, as I'm not very good in that to control these controls of the mobile because I'm still learning. Everyone should have to learn by the time of the death. So sorry for inconvenience. Any suggestions for improvement will be welcome very much because we are not born or trained these type of lectures, but I will appreciate your help to improve these lectures or anything that you should suggest me. Thank you very much.